This lecture begins our exploration of unhealthy microbial interactions. Here we introduce general concepts about unhealthy microbial interactions, and in subsequent lectures we will cover important pathogens. But wait, most people die from non-infectious disease. How important is it to learn about pathogens? Learning outcomes? Learning outcomes. So, the question is, the advances of modern medicine have made infectious disease globally rare. True or false? The global impact of infectious disease worldwide is significant. While over 70% of deaths are due to non-infectious disease, and we have come a long way since the 19th century, infectious diseases still account for far too many deaths. With the ongoing COVID-19 epidemic, I don't have to do much of a sales pitch this year to get you to understand the importance of learning about infectious disease. However, even after this pandemic is over, and it will be over, there are other infectious diseases we need to continue to fight. This series of lectures will cover some of the most significant infectious diseases. Before we begin, I want to cover a few key terms I will be using and make sure everyone is clear on their meanings. A pathogen is an infectious disease agent that causes damage to a host. A commensal pathogen is a member of the normal microbiota that may cause disease under some conditions. An opportunistic pathogen causes disease only in compromised hosts. For example, people who are immunocompromised, those with burns or wounds that have broken down the natural defenses of the body, or people suffering from malnutrition will have weakened immune systems, and then pathogens can infect them. A primary pathogen causes disease even in healthy people. However, even in these cases, a primary pathogen doesn't cause disease in all infected individuals. In other words, host susceptibility varies. There is also a range of where a pathogen falls, and sometimes they may be considered a commensal pathogen, but due to genetic changes, they slide into primary pathogen, or they may, by genetic changes or adaptations to the host, move from an opportunistic pathogen to a non-pathogen, etc. Today, it seems ridiculous to think that there was a time when humans didn't realize that microorganisms cause disease. But how did we figure that out? The germ theory of disease was a landmark accomplishment in medicine in the 19th century. The experiments of Lister and Pasteur led them both to suggest this idea. The work of Robert Koch on tuberculosis, anthrax, and cholera then gave strong proof that this theory was correct. For his work, he was awarded the Nobel Prize in 1905, and as part of this work, he developed Koch's postulates. Koch's postulates are the following. First, the suspected pathogen must be present in all cases of the disease and absent from healthy animals. Second, the suspected pathogen must be grown in pure culture, away from everything else. Third, cells from a pure culture of the suspected pathogen must cause disease in a healthy animal. And then finally, the suspected pathogen must be re-isolated and shown to be the same as the original. These postulates are still used today to determine the causative agent of infectious disease. There are caveats, however. Consider the following. Koch's postulates cannot be satisfied by... The correct answer is D. None of these choices will actually satisfy Koch's postulates. If you can't grow a pathogen in culture, you can't satisfy step 2 of Koch's postulates. If it's a commensal pathogen, it will be part of the normal flora of some people and not cause disease. If it's an opportunistic pathogen, it won't always cause disease in a host. None of them satisfy Koch's postulates, and you have to sometimes modify them as you think about it. There is a common set of steps pathogens follow as they infect a host. The steps are transmission from the reservoir to the host, attachment to the host, invasion of the host, and then host damage. Let's go through these steps. First, pathogens must carry out all these steps, and they particularly excel at attaching to specific sites, at invading into different tissues, and growing inside the host. Some pathogens produce toxins that aid them in these various steps. These toxins are secreted outside the bacterium, often using a secretion system, and the toxins themselves are often on pathogenicity islands. A pathogenicity island is a stretch of DNA that encodes genes involved in causing an infection. The loss of these genes makes the bacterium less infectious. Finally, some pathogens have the ability to invoke a strong inflammatory response, and this in itself can cause damage. So before we begin, let's look at each of these steps of infection. First of all pathogens have to maintain themselves somewhere and then be transmitted to a susceptible host. In these sites, pathogens remain viable and from which individuals can be infected. There are many different reservoirs for pathogens, and they are unique to each particular species that causes disease. Some are found in the environment, such as soil, Clostridium tetani, water, Vibrio cholera, listeria is found on plant material. Others can replicate in animals. The term zoonosis is used to distinguish these illnesses. Wild animals can transmit illness to humans. For example, COVID-19 and rabies were originally diseases of wild animals that have now adapted, in the case of COVID-19, to spread host to host. And domesticated animals. Campylobacter jejuni, a major cause of food infection, is transmitted from poultry. There are also illnesses whose only reservoir is humans. Successful pathogens will have a high carrier rate in these cases. Nearly 50% of humans carry Staphylococcus aureus. A carrier is a pathogen-infected individual who shows no sign of disease. To cause an infection, a bacterium has to be transmitted from its reservoir to its host. Transmission can occur in many different ways. Here are some examples. Inhalation of soil particles. For example, a person may inhale soil-borne fungi that cause respiratory infections, and this is the way blastomycosis is transmitted. Direct contact with a wound. Bacillus anthracis can infect wounds and cause infections this way. Consumption of contaminated water or food, or use of contaminated water for irrigation or food preparation. In many cases, this is how foodborne illnesses spread throughout the human population. Also, swimming or bathing in contaminated water can cause infection, and Giardia infections can begin this way. Transmission of pathogens can also occur from infected animals. Bites from a dog can transmit endocarditis, a serious systemic infection that affects your heart. Your mom was not wrong when she told you to never handle dead animals. They may be dead because of a disease, and that disease could be transmitted to you. Even light animals can transmit illnesses to humans. Contact with animal or human feces that may be in food and water can also be a source of disease. Also, sometimes foods are infected with pathogenic microbes that can then cause disease. There can also be indirect transmission through a vector. Most vectors are generally arthropods. For example, mosquitoes, lice, ticks, fleas, and flies. Pathogens that are restricted to humans must figure out a way to be transmitted from one human to another. This transmission can either be direct or indirect. The most widespread diseases are transmitted this way. Transmission can occur through direct contact via skin or sexual contact, or contact with other bodily fluids. If you look at this nice, lovely image of an unprotected sneeze, you can see how this person sneezing in your direction could spread droplets all over the place. Indirect transmission is also possible if a susceptible individual comes in contact with airborne droplets, fomites, or feces. Fomites are inanimate objects capable of transmitting disease. For example, the ability of COVID-19 to remain infectious for up to 24 hours on cardboard suggests it may be possible to get the illness from handling cardboard that an ill person has also touched. Once a pathogen has been transmitted to a host, it must then attach to its target. This attachment is specific. For example, enterohemorrhagic E. coli has a capsule that promotes adherence to the brush border of microvilli in the small intestine. As another example, Streptococcus pyogenes has an M protein that helps it bind to receptors on respiratory mucosa. Neisseria gonorrhea has pili that facilitate binding to the urogenital epithelium. As a final example, SARS-CoV-2, also known as COVID-19, recognizes the ACE2 angiotensin converting enzyme 2 receptor that is found on epithelial cells. These are very common in the lungs, and that is why this virus can cause infection there. Once a pathogen is attached to a host, it then may invade further. Invasion involves either entering epithelial cells or loosening the junctions between cells and moving into deeper tissues, or both. The transit to deeper tissues allows more widespread and significant infections. Invasion is not always necessary to cause disease. Some will stay at their original site of infection and cause their damage there. Those that do invade may be facultative or obligate intracellular parasites. The cell types colonized can be professional phagocytes, such as macrophages or other cell types. The picture at right shows Mycobacterium avium growing inside human cells. Host cell entry in these cases is usually an active microbe-directed process. The initial inoculum is usually too small to cause much damage in an infection. Thus, the bacteria needs to grow and replicate. Part of this process is to acquire nutrients. Extracellular pathogens will secrete hydrolytic enzymes that degrade tissues to release nutrients. Intracellular pathogens will obtain nutrients for the host cell cytoplasm. In both cases, there is a need for iron, which the immune system sequesters, as you learned about in Module 11. Many pathogens have methods of overcoming this and getting the iron that they need. Pathogens also need appropriate environmental conditions, temperature, pH, oxygen, etc. to grow, and in most cases the body provides this. Pathogens also have to sense and respond to the environment. The regulation of virulence factors, any function needed to cause disease, is often in response to a host environment, and quorum sensing often plays a role in this regulation. Finally, a pathogen causes damage to the host. Invasion of tissues and growth all by itself will cause damage, but these bacteria often also create factors that increase this damage. The production of toxins, either proteins or lipopolysaccharide, is toxic to cells. In the case of exotoxins, they often are targeted at specific immune cells to help protect the pathogen. Exotoxins are secreted, and they're usually proteins. Endotoxins are LPS, and they cause their damage by inducing the immune response, which causes damage. In some pathogens, secretion systems such as type 3 secretion systems will deliver toxins and other proteins directly into cells. Pathogens also produce hydrolytic enzymes to degrade surrounding tissues for invasion and nutrient acquisition. This also causes damage to the host. Formation or dissolution of blood clots can be a reaction to a pathogen against the host. Many of the times, these pathogen responses intend to disrupt a normal immune response. The advantage for the pathogens are that this damage often facilitates transmission to new hosts or to a reservoir. I want to dive into a bit more about the specifics of exotoxins because they are so important in many diseases. Exotoxins are proteins made by the pathogen that have a specific target in host cells. Exo refers to the fact that the toxins are secreted. In most cases, they'll be made up of at least two proteins and have an A/B structure. The B subunit is involved in binding to target cells and dictates where the toxin can enter, while the A subunit has the toxic activity. There are many different types of toxins and they often have a specific target. Because of this, toxins sometimes get names based upon the target they attack. Enterotoxins attack the GI tract, neurotoxins the nervous system, etc. Some specific examples of toxins include alpha toxin from Streptococcus pyogenes. This toxin inserts in the host membranes and creates a pore, and this can often lead to cell death. The classic strep test involves streaking a culture of the microbe from a throat swab onto blood agar plates. If a zone of clearing develops caused by alpha toxin, it is a strong indication you have a strep infection. Shiga toxin is a classic AB toxin, and we will talk more about it when we cover E. coli infections. Stable toxin, another E. coli toxin, uses a third common mechanism. It activates membrane-bound guanylate cyclase, which is a common signaling pathway in host cells. This causes an accumulation of cyclic GMP and the loss of chloride ions. Water then, because of this, enters the intestinal lumen, causing diarrhea. Target delivery of effector proteins can also alter cellular function. And in these cases, what happens is the organism, in this case Salmonella, will bind to the membrane, and it uses a type 3 secretion system to deliver toxins and effector proteins directly into host cells. These effectors can have many different functions, and I'll talk about specific examples as we go through various diseases. One of the other things that you have to do if you're infecting a host is avoid the host's immune response. As you learned from your study of the immune system, hosts have many different defenses to try and counteract and inactivate pathogens. Some of the things that pathogens can do in response include avoid or hide from the immune system. They can do this by growing inside cells, intracellular growth, invading privileged areas not monitored by the immune system. Sometimes joints can be an area, or deep inside bone tissue can be very difficult for the immune system to reach. They can avoid or inhibit phagocytosis. They could also lack microbe-associated molecular patterns, MAMPs, for detection, or they will mask these. They can also actively disrupt the immune system. They can kill immune cells using toxins. They can interfere with immune signaling. Maybe they degrade some of the cytokines that the immune system produces. They can interfere with immune effector function. Maybe they inactivate or bind antibodies in an unusual way. Maybe they counteract complement. For example, Bacillus anthracis creates a capsule. This capsule will hide some of its MAMPs. It also has two toxins that neutralize the immune system. Finally, I want to end this section by emphasizing that the ability to cause disease is multifactorial. Each virulent trait helps a pathogen infect and survive in a host, with some being more essential to cause disease than others. Shown here is the array of factors that allow Salmonella species to cause gastrointestinal illness in humans. I don't want you to memorize all these different factors. It just illustrates the point. We will end with a few clicker questions. A primary pathogen is? And finally, which of the following is a known method of transmission of disease? Okay. A primary pathogen is a microbe that causes disease even in healthy people. So the correct answer is C. Which of the following is a known method of transmission of disease? All of the above are known methods of transmission of disease. The rest of the lectures in Module 12 will cover specific illnesses, and they will be organized into individual movies about each illness.